Welcome to this week's molecular biology. So, this week we're going to talk about molecular biology. What I want you to pay attention to is the chapter is relatively dry. It just talks about the flow of genetic information. But there's really more to it, and I want you to think a little bit about that. So, we're going to look at chapter 9, read chapter 9. And then we'll talk about what this means that we've just learned here. So, background information, we're just going to talk about the structure of DNA, transcription, translation, and the role of DNA. You can watch the transcription, translation materials here. There's a couple of things here for you to look at. There's a lot, so just pick and choose what you want. Your assignment material are there's also a little bit on molecular biology and on biotechnology for those of you who want to read up a little bit more on that. But look at the transcription translation video down here. When you're looking at the chapter, I'm just going to pop into the chapter real quick. So the chapter talks very quickly about the kinds of things that we can do. So it's the genetic material, obviously, that's passed along. And Dolly was the first sheep that we made that was a cloned mammal. So um, the chapter brings up this idea that we can manipulate this, right? Once we understand how something works, we can manipulate it. Um, so we're going to learn about how this works. We're going to talk about structure of DNA. We're going to talk about replication, transcription, and translation. And I'm going to jump into translation just real quick. Actually, I'm going to jump into transcription first. Sorry. There's the central dogma that says that DNA goes to RNA, goes to protein. That's true, except in case of viruses, because viruses sometimes they come in and they bring RNA. And when viruses bring in RNA, they have to make it into DNA before they can make it into protein. But so the central dogma is true for us. It's not necessarily true for everybody. Translation is basically you're taking nucleic acids and you're turning it into there's a secret code how you can turn nucleic acids into protein, and the decoder that does this is this protein called this thing called tRNA. um, tRNA literally is your decoder. It has the anticodon on one side and the amino acid on the other, and it recognizes the anticodon and then allows you to put the amino acids together. Gene regulation is something that happens differently in eukaryotic and prokaryotic cells. And this is important when we try to modify genes, because prokaryotes, we've trained many bacteria to do our bidding when it comes to creating, for example, things like insulin. Um, Insulin, people who have diabetes, they used to have to rely on insulin literally from pigs, and lots of pigs were slaughtered for their insulin. Nowadays, we can do this with bacteria, and bacteria make this stuff for us. And so we had to learn how bacteria control things, how we can get bacteria to do the things that they need to do. Um, the book doesn't very much talk about mutations. Um, but mutations are, those are built in to the system because both in transcription and translation, there are mistakes that can happen. So, for example, when you're transcribing a piece of DNA, if the sequence that you're transcribing is not correct, you end up with a different codon, And then when you're translating, if this is what you're supposed to be making, right? Let's say, for example, you're supposed, like in sickle cell anemia, for example, there is a sequence that is, it is, it's supposed to be GAG, a glutamine, and in sickle cell anemia, that's changed into GUA, which is a valine. Um, so what happens when there are mutation? When there are mutations, sometimes there are diseases. Sometimes mutations are devastating, and they completely destroy the protein. Um, and those are the kinds of things I want you to think about today, as we're thinking about what we're doing here. So that's the reading. In the demonstrations, I've got a couple of different things for you. We're talking about protein synthesis in a, in a more fun way. But then I want you to think about genetically modified organisms, kind of like Dolly, the sheep. We There's been lots of discussions in the public, lots of ideas in the public about what genetically modified organisms are. And I want you to think about a little bit what they do. Read up on them. Google genetically modified organisms. Um, I've got a specific one that I want you to read up on, BT corn. Um, BT corn is a type of corn that has a resistance gene built in. And I want you to think about what that means. You probably have heard of Roundup Ready corn, which is the stuff that is resistant to Roundup. And there may be a difference in what those two things mean. How are they different? How are they better or worse? So that's the thing about GMOs. And the next thing I want you to think about is mutations. Um, if we can modify the DNA, or if we can, first of all, if we can recognize mutations, would you want to know? Would you want to know if you have the gene for Huntington's? We can find the DNA. What would it mean for you to have this? And there's a movie here, The Gene by Ken Burns. It's quite long. It's 4 hours long. So I'm not expecting you to read to do the whole thing. Um, but I'm asking you to so it's quite entertaining. So you may just watch it just to be watching it. But this old woman thinks it's quite entertaining. You probably don't think so. But so there's a couple of there's about 10 minutes here at the end, and then there's about 10 minutes here that tell you about two different diseases and two different things. Um, and I want you to kind of watch at least those sections, and talk to each other. Talk to each other about what you think. And then the assignments are going to be relatively short and sweet, I think. Um, you go to the Chapter 9 quiz, and then after the Chapter 9 quiz, you get to the GMO quiz, which just asks you about watch the GMO videos that I gave you, and then think about what are the things, how did they benefit us? Look some things up if you're curious about some GMOs. Like a genetic, like for example, this bacterium that makes insulin for us. That's a genetically modified organism. um, the bacteria that help us make vaccines. Those are genetically modified organisms. But also the flavor right tomato, that's also a genetically modified organism. So where, what's a good thing, what's a bad thing? Just think about it, talk about it, and write about it semi-intelligently. I just want you to have a little bit of a knowledge of this. And then the last but not least of the assignments is simply an opinion thing. Would you want to know if you have Alzheimer's disease? What if, like in the case of this, when you listen to the gene, when you watch the gene video, this gentleman didn't realize that he had the possibility to have Huntington's disease, because Huntington's disease is a nasty disease. It'll kill you. It'll kill you as an adult, though, after you have children. And this guy found out that he was potentially a carrier of the gene after he had children, when his sister figured out that she carried the gene. So then he knew that the gene was in his family, and he had to make a decision whether he wanted to know. And the fact of the matter was, he didn't. He didn't want to know, because if he knew, or if he had it, then there was a possibility that he had passed it on to his children, and he just didn't want to know. Sadly, he had it, and he didn't get tested until he started to get symptoms. So, what do you think? Would you want to know? Would you not want to know? Would it make a difference if there's a treatment for the disease, or if there's not a treatment for the disease? Those are the kinds of ethical questions and, personal questions you got to sometimes ask yourself about. And a lot of times, biology and ethics are actually very closely related to each other, because we can do a lot of things, but we probably necessarily shouldn't do them. Case in point, there was a story in the news 2018, 2019, where a guy in China um, modified the DNA of two little girls. That he literally made a genetically modified human. Um, this guy's now in jail, because the scientific community said, no, we can't do that. Because when you make a genetically modified human, there's so many things we don't know yet. If you're genetically modifying a human, that genetics is going to be passed on forever. Those genes are going to go on to their children and their children's children, and it doesn't stop with just that one person. Um, and so that's a real problem with humans. So, yeah, so think about those things. Think about the implications. If you're interested in the next chapter, we don't actually cover it because biotechnology is beyond the scope of this class. But um, there's more about biotechnology, genetic engineering, those kinds of things in that chapter. And that's all I have for you this week.